biotechnology, about biotechnology for a sustainable bioeconomy. We are really happy to have you all here. My name is Caroline. I am part of IGEM Design League team, and I talk to you from Brazil. Invited here today industry experts to talk about biotechnology, what they are doing about today for a sustainable bioeconomy, and what are their perspectives for the future of bioeconomy in Latin America, but also in a global perspective. I would like to talk a little bit about our partners. So Ginkgo Bioworks is an American company that uses synthetic biology to design, develop, and optimize products for different applications, such as key ingredients for vaccines, crops, plant-based meat, and so on. We have Instituto Senai de Inovação, or Senai Institute, Innovation Institute for Biosynthetic, which is a Brazilian organization that develops more sustainable products and processes through chemistry and industrial biotechnology. They have a team of experts in the field of biotechnology, chemical transformation, process engineering, and fibers to provide services for other industries, not only Brazil, but also globally. We have also Vale Institute of Technology for Sustainable Development, which is a private nonprofit institution for science, technology, and innovation. They develop projects seeking better options for the present and for the future, promoting technological solutions and social environmental transformations. They are located in Belém, in Brazil, in the surroundings of Amazon forest. Guilherme Oliveira, one of the experts invited here, he works at Vale. Well, this event is organized by IGEM Design League, which is part of IGEM Foundation. IGEM Foundation is an independent nonprofit organization dedicated to the advancement of synthetic biology field. IGEM has several projects, and it organizes the largest global students competition on biotechnology. Last year, we had more than 350 projects competing in the global competition from over 40 countries in the world and more than 7,000 students. It's a lot of people.
Bien. Sí, estoy en call.
Okay, yeah, someone was speaking. Well, one of the main goals of IGEM is to empower local people to solve local problems using a synthetic biology framework. But we know that every place is different. Every country has its own challenges. So it's not the same experience for every country and every region. That's why IGEM has a new purpose, that is to make sure that the synthetic biology field and all its power gets developed by everyone everywhere. And this is how League's program was born. And the IGEM Design League is one regional competition for Latin American students to develop synthetic biology projects focused only on the step of design. So the main goal of this competition that exists, this is the third year, the main goal is to help Latinos to kickstart their career in synthetic biology by providing a competition, so challenges to push them forward, and also educational tools such as IGEM Academy, which is a platform with more than 25 hours of content about synthetic biology in Portuguese and Spanish. And we provide this competition in order to train and to prepare the next generation of talents for the future bioeconomy. Our main goal here in iGEM Design League is not only accelerate education in synthetic biology, but also accelerate synthetic biology in Latin America. It means that we need to engage not only students and teachers in the competition, but we need to connect and engage with other stakeholders such as industry, public agencies, entrepreneurs, startups, companies, and everyone that is interested in the field to connect everyone and empower our region. We know that Latin America is internationally recognized as a biodiversity hub, and this is really interesting because this is a very rich resource that we have in our hands that we could use and apply biotechnology and synthetic biology to sustainably explore this biodiversity without having without losing biodiversity, so keeping the genetic richness. And this is not new. The governments are setting goals, very bold goals for the future, aiming to reach a sustainable bioeconomy. So Costa Rica has launched its own national bioeconomy strategy in the past few years. Brazil has a public policy for biofuels and also for biotechnology for the past 15 years, I think. And also last week, this is new, the US government set a very bold goal for bioeconomy, and it includes a target for 30% of bio-based chemicals in 20 years. I think it's 20 years. So we have a huge opportunity here to use biotechnology tools to develop a new and more sustainable bioeconomy in the future. And that's why we are here today. We want to talk about experts from the field and discover how we can apply biotechnology for a sustainable development and what they think what they are doing today and what is the future of this field. So thank you, Daniel, Marilene, and Guilherme for accepting our invitation. And now I would like to invite Daniel to introduce himself. Daniel from iGEM. We have two Daniels here today. And also to start the roundtable. Thank you, everyone.
It is a very good name. Thank you, Carol, for the introduction. You gave us really good insights to start the conversation. And thanks everyone for joining this roundtable. This is the first of many events that we will have during the year to discuss on the applications and the areas of impact of synthetic biology and how biotechnology and synthetic biology are shaping this new bioeconomy. And in this case, we're talking about sustainability and how to create a more sustainable and green future for everyone and everywhere. So here we have three incredible panelists with us, working in very different companies, all of them doing some really exciting biotechnology research and development. And I want to start precisely talking about that. So the first question for all of you is, what are your companies doing in the biotechnology and synthetic biology field? What are your main projects? So talk a bit about yourself. Please introduce yourself as well. Tell us who you are, what are you doing in these companies, and enlighten us with the information about your companies. And I will start with Marilene. By the way, congratulations on your new role. So please tell us a bit more about you and your company.
There it is. Thank you for having me today. Thank you for congratulating me. And yeah, so my name is Marilene. You can call me Mary. I work in a company called Lanzatech. We are here in Chicago area in the US. I'm originally from Brazil, actually, and I'm a biologist. I moved to the US in 2016 to work as a research fellow at Boston University. And I've been working for my whole career in metabolic engineering, synthetic biology, automation. And when I moved to Lanzatech was to work again in metabolic engineering, synthetic biology, but mainly in automation. And then recently, as Daniel mentioned, I moved to a new position as more focused to work in synthetic biology strategy. And what does it mean? It means that I'll be more involved with the technical and economical aspects and sustainable aspects of what we produce, and in the prioritization of what we do in the synthetic biology lab in terms of molecules, materials, and partnerships and whatnot. At Lanzatech, we use synthetic biology to modify our proprietary microorganism, which is a naturally producer of ethanol. We have 2 commercial plants, or 3 now, in China, and we are deploying another one in Europe with ArcelorMittal. So this microorganism is anaerobic, and it uses, instead of the carbon coming from sugar, for example, it uses carbon and energy coming from CO and CO2 to ferment and produce ethanol. So our group specifically is invested in modify genetically and in high scale, high throughput, this microorganism to produce other things other than ethanol. So that's our major goal now. The group is divided between the different areas like strain engineering, automation, enzymology, and each one of these subgroups, let's say, it's focused on the same goal, which is modifying effectively this microorganism. So that's mainly what we do here and what I do here.
Thanks a lot, Marilene. It's really interesting the work that you're doing in Lanzatech, and I think this is one of the key factors for the future. Bioeconomy, for the ones that are not familiar with the term bioeconomy, by the way, you can start writing your questions in the chat box. This relates to the economic model of using biological resources and natural resources to basically produce anything, right? But in the way that we normally understand bioeconomy, and especially here in Latin America, where we have a lot of biodiversity and a very rich set of genetic resources, we normally tend to think of it as something that we can exploit. And this is a very interesting point for the future, because synthetic biology wants to get rid of that term forever. So we're not going to exploit anything. We're going to create sustainable sources of molecules and resources. And one of my favorite parts of the work in Lanzatech is that they are bioremediating the air, capturing carbon, capturing CO2, and feeding microorganisms with CO2 to produce really valuable molecules. So this is a really interesting way to turn a problem into a solution, and also creating a new generation of bioeconomy products. So this is something to consider. So let's take some notes on this work, and some related work also creating a very new way to produce products. And it is very tied to the bold goal set by the US government last week is the work of Braskem. That is a Brazilian company that now is starting to invest heavily in research and development in the US. So my tocayo is the word we use to refer to someone with the same name in Spanish. So Dan, welcome, and please tell us about you and the work that you're doing in Braskem in the US.
Certainly. So my name is Dan MacEachran. To make it easy, I'm Dan, and I guess you're Daniel. So that'll ease things a little bit. So hello everyone. I really appreciate the opportunity to come and speak today and be part of such an august sort of group of individuals. So thank you very much. So yeah, my role at Braskem. So I'm the head of biotechnology at Braskem. My background's in biotech. Before joining Braskem, I worked for, at that time, what was a small startup, which was Greenlight Biosciences. It's now a publicly traded company. I think they launched at a valuation of something like $2 billion. I worked as the head of R&D there, and led one of the teams that developed their RNA technology, which is really the cornerstone of that company today. But I would say more broadly, we were focused on using cell-free biology, which is another sort of area of biotechnology that I think is a really promising and interesting one. Before that, I worked for a Department of Defense contractor focused on making biofuels. So I've worked in a couple of different spaces, biofuels and ag chemicals, nutraceuticals, and now I'm in the petrochemical space. So my role at Braskem, I wear a couple different hats. So as I said, I'm the head of biotechnology here. I'm responsible for managing a team of scientists and engineers. We have about 50 folks in our biotech group today. I would say the majority of those, about 45 of them, are based in Campinas, Brazil, at our R&D lab there. And then we have another 4 or 5 folks here based in Boston. Boston is where I'm based out of. You know, I think Braskem itself, right, we're a petrochemical company. We're based in Sao Paulo, Brazil, but we have industrial assets and offices pretty much everywhere in the world. We are one of the world's largest producers of polyolefins. So if you go and look on the bottom of a cup or something and you see I think it's a 2, 4, or 5, that's something we've made. And if it's got a 3, we probably made that too. And so, you know, in all aspects, we're a very traditional petrochemical company. But I think the thing that differentiates us from our peers is that really from our inception, we have had a hard focus on sustainability. And I think that the thing that we all like to point to quite a bit there is our one of our major products today, which is our green polyethylene. So Braskem in 2010 commissioned our green polyethylene plant based in Triunfo, Brazil, which currently produces about 200,000 tons of renewable ethylene every year, right? So what we do is we take sugarcane that's fermented to ethanol, we procure that ethanol, and then chemically convert that to ethylene, and that's the same ethylene that we make from petro sources. What's really cool, and the story I love to tell about that, and the thing that gets me really geeked out and out of bed in the morning, is that for every ton of polyethylene we produce, we either fix or offset a total of 5 tons of carbon dioxide, right? So that one plant offsets or fixes a total of 1 million tons of carbon dioxide per year, and we've been doing that for 12 years. We're currently working to debottleneck that plant, expand the capacity to allow us to produce more there. We're also looking at building plants in Asia as well as North America today. And those are large efforts on our part. Not necessarily a biotech technology, although there is a biotech arm to it. So in addition to that, we have a real big focus on renewables, right? So some of you may be aware that Braskem made a commitment to be carbon neutral by 2050. And a key part of our success and part of our strategy to do that is through the production of renewable chemicals. And we approach it very much from a holistic viewpoint, right? We're very agnostic as far as the technology that gets us there. We just want to go from renewable feedstocks, whether that's sugars, plant oils, lignocellulosic biomass, and in some cases, even things like CO2 and methane, and convert those to both drop-in products as well as new products, new to the world products. And again, we don't really care what's in the middle, right? It can be catalysis, it can be biotech, it can be thermochemical, or it can be combinations thereof. And that's really sort of our key focus today. So where does biotech live within all that? Obviously, we've got a huge focus on making a variety of different chemicals and materials, both both drop-in chemicals and materials, as well as new chemicals and materials. So I think I've probably gone over my time a little bit, but that's a general sense of me and Braskem and what we do.
Thank you, Dan. And this is also very interesting because this is another key factor for a sustainable bioeconomy. How can we make the transition or reduce our dependence on the petrochemical industry? And how can we transition from a petroeconomy to a bioeconomy? And Braskem is one of the companies leading the way and leading the development of technologies from Latin America, and also creating all these new tools to produce these valuable chemicals. Another key factor of this transition to a sustainable bioeconomy is how can we preserve our biodiversity and how can we make a better use of our biological resources, all the richness that we have in our ecosystems and in our region. And Instituto Tecnológico Vale is doing an amazing work also on supporting research across Brazil to allow this and to enable these new technologies to happen. So here we have Guilherme that is leading that institute. So welcome, Guilherme. Tell us a bit more about yourself and the work that you're doing and your mission in the Valley.
Thank you, Danielle, and thank you, Caroline. And it's a pleasure to be here with Dan and Mary. Very interested in hear what you have to say. And thanks all of you people who are here to help us in this discussion. So my background is in genetics and genomics and bioinformatics. And in my prior life, I worked with human infectious diseases. And for the last 8 years, I moved to the Vale Institute of Technology, initially to head the genomics group, to study biodiversity, applying those tools. And now I head the institute for the last 2 years. And what we do here is try to understand the environment, both physical and biological, initially where Vale operates, to help the company fill knowledge gaps that will contribute to preserving nature and contribute to society in those territories. So we have conducted an extensive amount of research in these areas. And for the last year, we have expanded this research to an Amazon-wide perspective for 2 reasons. First, because by the fact that we are in the Amazon, we are one of the largest companies in the country with a global presence. The company feels that it needs to extrapolate all of this work that was done in the Carajás region, which is now the heart of the company in terms of mineral production, to the Amazon, and also translate that to the benefit of the people who live in the region, not only because we value biodiversity preserved as it is. I think everyone here is more than happy to see it preserved, but we need to consider the people that more than 25 million people that live in the Amazon that deserve a better life. And what we have to do with it is to transform this biodiversity into real resource to these people while maintaining the forest standing and the rivers flowing. So that's the challenge that obviously we will not get there by ourselves. We need everyone involved in this transition. But we are doing a significant amount of work there. And so why are we here? The reason is that we all understand there's a large amount of the genetic resource contained in the forest. I think everyone agrees is phenomenal, but it has not been revealed. So it's phenomenal, but it's being used sometimes illegally. But on the legal ways that it's being used very little and for products with very low added value. And the way that we think that we can transform this is by revealing the genetic resource that's still hidden in the forest. And one of the ways to do that is through synthetic biology. So we are here as a research, we are a private nonprofit research institute, and we operate very much as an academic institution. So we don't produce anything except for science, which is all right. But I think it's very interesting that we have industry present in this discussion, because obviously the academic research is absolutely essential for everything that we'll be discussing. But without industry, we will have a hard time to translate that into actual value for the forest, for the traditional people in the forest, and for everybody else. I think the impact will be global. And I think that the way to do this, these two companies that are present here have a role, but I think the young folks that are present in this discussion are the ones who are really going to take the bull by the horn and make this happen. So we see ourselves as one little engine there to help in this movement. So thank you so much for the opportunity to be here with you.
Thank you, Guilherme, and thank you for the support for this initiative. And actually, regarding this last part, this is precisely one of the objectives of this series of panels, because we're going to create a dialogue between the young population that is part of IGEM and it's engaging with synthetic biology, with the industry, because we believe that we don't need to convince the academic part of the region that we need to be aware of the potential of biotechnology. But we need to start talking with other types of companies and with other sectors of industry, so they can engage and they can collaborate with companies like yours to start working on this transition to a more sustainable bioeconomy. Well, now that we've finished this first round of introductions, it's curious because we have a very Brazilian panel today, and we have also a very global audience. I saw people saying hi from Dominican Republic, from Guatemala, from Colombia, from Bolivia. I'm seeing people from India, from Europe, from Lebanon. So welcome everyone, bienvenidos. And yeah, I think we should start the discussion, setting the field and talking about the technologies for the future. Dan mentioned something really interesting about their company, that is they are agnostic. In IGEM, we talk about a lot about synthetic biology, but the reality is that we need all available technologies to make this happen. And this is something that also Lanzatech is doing, not just using synthetic biology, but also other technologies. So the question for all of you, first to start, is what are the most exciting technologies that you are working on right now, and what do you think is the future for these emerging biotechnologies, and how can they contribute to reach this sustainability? And I will start in the other way around. So let's go first with Guilherme. What do you think about that?
Thanks, Danielle. And I think we have complementary views that complement each other on the technologies. One that I think is very important for us, and I think somehow taken for granted, as in the way that it's generally thought to have been commoditized, but I hope I can demonstrate it hasn't, is genomics. Because in the region, without doing proper genomics, without knowing how to handle this technology and producing it locally, we'll never do that. You can see, you look at the global projects that are out there, none of them are south of the equator. And it's not going to happen. I mean, the rich countries are happy doing, and I'm not against it, but they're happy to do a butterfly in some island that has been that's been okay. And we have the issues in the Amazon that I don't need to remind you of what's happening. And so the issue is here in the south. I mean, Europe has been re-greening for a while now. So we need to develop these tools, but we can use all of the help that we can get from the countries and the labs that have been doing this for a while. But we need to do this locally, and we need to own, in the sense that we own the technology, we know what we are doing, can manage it, because without it, we won't be able to move forward with the hardcore synthetic biology that Mari and Dan are already deploying in their activities. And one of the companies that sponsors this event. So I think this is the first step for countries in the developing world to make sure that they have a significant grasp on the technology. And what I can say in that regard is that we have launched with the environmental office in Brazil a very large project in that sense. So to sequence genomes of plants, animals, and microorganisms with a focus in the Amazon, but not exclusively, and in connection with a large network of labs. So I think it is significant, but it's a first step in that direction, and I think we need to do more. So I invite other companies to join us in this effort. But this is not the end. Only doing genomics that doesn't get us there. It's the way forward. So I think I'll stop here, because I'm sure that Dan and Mari will have plenty to say regarding the next steps.
This is something that I will also highlight, like we need to create technologies with a Latin American perspective, right? Like with this new identity that not just using technologies, but also developing our own technologies. So let's go with Dan. Tell us about your perspective from Braskem.
I love the order here, because I think Guilherme spelled it out perfectly, where it's sort of, we have the foundation, right? And it's all information at the end of the day. And that information flows from genomics and then upwards, from there, and enriches all the things we try and do in synthetic biology. I think for me, I think sort of starting high level, one of the things that I'm really geeked out about, and again, that's like a major focus for my company and for my group, is how do we start to push together the disparate disciplines, right? How do we take these disciplines like process technology, like catalysis, biotechnology, all these things, and start to push them together, right? Because my general sense is, for the most part, we don't have the technologies today we need to get to where we need to solve some of the big issues we have around circularity, around carbon emissions, all these kind of things. It's going to take pushing all these different parts together to develop entirely new technologies. So I think one of the things that I'm most excited about is, how do we start to push those together? How do we have processes that start with biotech and end with catalysis, or vice versa, to really enable us to get to the types of chemicals and materials that we want to drive forward into the marketplace, and really drive us towards our sustainability goals as a community. I think the other thing I mentioned before, getting a little more specific, cell-free biology to me is a massively underutilized tool. And I think from a lot of different perspectives. I think both from a sort of just a pathway sort of validation tool. And this is something I think Lanzatech actually had a beautiful paper together with Mike Jewett and a couple others where they use that as a tool. And to me, it's a massively underutilized tool. I think it's something that my team is going to continue to leverage more and more and more as we go forward. Just how do you prototype pathways very quickly and then encode those back into a cell? But I think also more broadly, from a production platform standpoint, I think cell-free has a ton of potential advantages over standard fermentation technologies. And I think we're just really starting to scratch the surface of that. Certainly Greenlight Biosciences was a pioneer in this space. They've since gone a slightly different direction with their cell-free, but there's a bunch of new companies that are now coming online, starting to relook at some of these things. So I think those are two big ones, and maybe just one last one, that, not to use up too much time, but one of the other things that gets me really excited is is the expansion we're seeing in chassis organisms, right? So when I was coming up, it was, you know, if you want to engineer a bug, it was either E. coli or it was Saccharomyces. Both great bugs, both have their utility, but I'm a big fan of the model of pick your chassis based off the molecule you're making, right? So Lanzatech is working with a bug today that, you know, 10 years ago, 15 years ago, people would have said it's unengineerable, right? And now it's expanding very quickly. And so I love this concept of, hey, today I want to go make a solvent. So I'm going to go focus on a solvent tolerant bug. I'm going to grab a Pseudomonad. You know, hey, I want to make something that comes off acetyl-CoA. You know what? This bug over here is great for that. That concept of picking it correctly. And I think, again, we're at a point now where engineering these bugs, the tools that are there are sort of solving that question, right? Of can we engineer this and opening up a whole world of new possibilities. So those are the three things that I'm most sort of excited about in our field.
This is a very interesting point, this last one. When I was a student and I participated in the IGM competition, I just had those two options. And my team was like, no, we want to create a new chassis and we want to work with this photosynthetic bacteria. It was a very good choice, but a very bad idea because it's super hard. We just didn't have the tools. And now companies like Cultivarium is developing platforms and tools to work with more and more non-model organisms. And now the tool set that we have is very big, and I think it's opening a lot of possibilities to create. And let's close this round with Marilene. What do you think?
Yeah. So I agree with Dan about cell-free technologies or everything that can speed up our process and make it faster, more scalable, and also that can decrease costs. Because yeah, it's so hard to make these microorganisms to produce whatever we want, and especially if they are not natural producers, you need such a crazy amount of modifications and tests and DBTL cycles, design, build, test, and learn cycles to get to the optimal producer, that everything that can speed up the process and make it less costly for us is very interesting. So yeah, for me, microfluidics and cell-free and automation just in general are great and powerful tools to get us there faster, I would say. Another thing is that with this automation and scalability, it comes more data to analyze. And this is something that is really important for us. Since synthetic biology up to the commercial plant, we collect a lot of data every day from all aspects of production. And to analyze this is such a thing that we cannot do by ourselves. So machine learning and AI just in general, or computational tools in general, are something that we invest a lot just to get our work smarter, let's put this way, just to be able to analyze and come up to some conclusions more easily. We have this dedicated team for this as well. In terms of the company in general, we also want to be agnostic in terms of the feedstock. So we are investing a lot in gasification, gasifiers kind of technology. We demonstrated already in Japan with municipal solid waste that we can gasify this waste and produce the synthetic gas rich in CO and H2 that our microorganism loves more. So in China, for example, these commercial plants are capturing CO and CO2 coming from the chimneys from a steel mill that are pumped in these fermenters. And so but we want to use other waste streams as well. Like, for example, depending on the location, we can use municipal solid waste, we can use agriculture waste, or cement plants. So we want to be also agnostic in terms of from where this carbon is coming, and to truly create the circular economy that we are aiming to. So I don't know, let's say you have ethanol and you produce plastic from this ethanol. Then what's going to happen with this plastic? So we want to be able to gasify the plastic that is not recyclable to produce more ethanol and close the loop this way. So gasification is something, for us, it's also very important to couple synthetic biology with engineering since the get-go. Just because scale up, it's a very hard process to get efficiently. So since we started the synthetic biology group to modify these organisms to target these molecules, the engineering guys are always included as well to help us figure what kind of fermenter, what kind of equipment we are going to need in the future to scale up the process. So these are the main things I would say that we have been more focused on.
This is a really great list of technologies and trends to have into consideration. And I think right now we're living a very interesting moment in the biotech field, because a lot of things are happening. We are always talking also about the intersection of digital technologies, automation and biology, and it's incredible how every week things are changing and evolving, and we've seen a lot of new advances in this field and in every single technology that you just mentioned. And we're also in a very interesting moment, because after the pandemic, a lot of people started to pay attention to that technology, right? Some of that people is now in the government, and particularly the US government have been very keen on promoting this transition to a bioeconomy. So let's go a bit deeper on your work, and I will start with Dan and Braskem, because one of the bold goals that the government defined last week, I think some of those goals are more bold. Nobody said realistic, but some are like very visionary, I would say. But there is one particular that I think we could achieve on time, and I say we as humanity and not just the US as a country, and it's replace 90% of the plastics used in the US for bioplastics or plastics produced from natural renewable sources. Obviously, Braskem is doing a great work, and they have a complete history in this. So tell us, Dan, what do you think about this bold goal, and also what are the challenges for this? Why are bioplastics still not widely used with the technology that we currently have?
Yeah, I would say, personally, I'm very excited by the announcement. I think, I have two small kids. I enjoy doing sports, which are very based on, I love to ski, I love to scuba dive, and want to share those with my kids. And so, anything we can do to drive carbon negative technologies forward that will ensure that they continue to be able to do those things in the future, and everything else with it, I think is very exciting from a personal perspective. From a professional perspective, we're also very excited by the enthusiasm I think we're seeing from the U.S. federal government, as well as what we're seeing in Europe around bio-based products. As far as what has held them back, I think to do that you need to sort of look at bio-based products, bio-based materials, and sort of divide them into two categories, right? In my opinion, there's bio-based drop-in products, So these are things like the green polyethylene that we produce today. It would be green polypropylene. You see a lot of news about green PET. Braskem has technology to produce one of the co-monomers for that, but then you've got the issue with the other one. And then there's the non-drop-ins, right? These are entirely new materials. And I would argue a lot of the biodegradables we think of probably live in that later category. So I would sort of separate them a little bit, and then we can dive in a little bit more. I think for the bio-based drop-ins, I mean, the classic issue is always one of cost, right? You know, we've got an industry, the standard petrochemical industry, that's been in place for 70 plus years. There's a lot of work that has gone into making those processes as efficient as possible, right? When we think efficiency on the petrochemical side, it's 0.01% improvement in efficiency is, oh, everyone's excited by it. So I think you're up against that incumbency to some degree. I think there's also on the incumbency side just a lot of spent capital. You know, people have put a lot of investments into their plants, et cetera, and we're up against that. When we go to build a new renewable drop-in chemical or material, you know, you either have to find ways to use existing assets, which was the case with our green PE, right? We built it in a plant where we can take that green ethylene and feed it into the same polymerization units we've been using for all these other petro products. So you've got to find ways to do that, and or you're going to have a major capital investment that you're going to have to stand up against, you know, that sunken capital, and that's going to drive your economics. Right now, how do you get around that? Well, one issue is you got to do a better job of selling the value proposition, right? So again, when we first started with green PE, and this is again me speaking from, it's a personal opinion, not necessarily the company line, it was a tough road at first. When we took that out to the marketplace, people said, well, why should I care about this? And what we really ended up having to do was go further in the value chain. We had to go to the brand owners, and we had to go to the customers and say, is this something you're interested in? If so, ask and pull back up that chain, and we'll start pushing it. So I think it forces us to change how we think about things. I think one of the other issues that we've seen, and I think other sort of companies as they're bringing drop-ins into the marketplace is this concept of sole source. So today in the marketplace, most formulators, those are the people that buy our plastic and turn it to the products that we all use, they like to have multiple sources of the material that they're buying. And the reason they want to have that is it's a hedge, right? They want to make sure they're getting the best price, but they also want to make sure they have a reliable source of material. When I come in and I've got my fancy new process to make, let's say, green propylene, green polypropylene, I'm the only producer. And that makes the value chain a little bit nervous. And so this is actually something I've been pushing the Department of Energy to think about quite a bit, is how do they start to enable getting around some of these sole source issues? Are there ways to have cross-licensing agreements, et cetera, where companies can come in? It's sort of a boring answer to why we have some of the issues, but it's a legitimate one, and one that you see pretty broadly. On the new materials side, it's a little bit different. There's some of the same things. Obviously, cost is always going to be a part of it. Obviously, sole sourcing is going to be a part of it. But there's, again, this incumbency, but it's a little bit different. You know, again, if we look at the petrochemical industry, the plastics industry, they've spent 70 plus years, you know, maximizing profits, figuring out how to take ethylene and propylene and a couple other key chemicals and make a whole pile of products out of those. They've invested in processing equipment. They've invested in everything you can imagine. So now you bring a new material into the value chain, and you say to the formulator, the person downstream of you, say, well, here's this new product. And they say, well, geez, last year I spent $1 billion on this processing equipment that's good for PE, but I don't think it'll be good for your product. And so, you know, it's hard for me to justify that investment. So I think that's one issue that you run into is the incumbency. I think the other thing that I've seen a lot of is being able to link the synthetic biologist who says, I've got this cool molecule I can make, and the actual product at the end of the day, right? It seems like it should be pretty straightforward, but there's a bunch of leaps in there that are really difficult. And so how do you, again, get from here to here when you're talking new materials and new biomaterials, I think is a huge challenge. I think what we're starting to see is there are better tools coming online, things that are driven by artificial intelligence, machine learning, things like this, that start to say, okay, can you either retrosynthetically say, well, here's a characteristic and let's work backwards and find a molecule, or working forwards and say, hey, I've got this molecule, I think it makes this polymer, and I think this is how it's going to behave. So I think those types of tools are starting to come online that are making that easier. They're far from being, ready for the big game, but they're definitely coming along. I think, again, the other thing, and I'm going to beat on this again and again and again, is forcing those teams to talk to each other, forcing the polymer scientists of the world to talk to the synthetic biologists of the world and say, hey, here's what's interesting and here's why. A synthetic biologist chewing on it and saying, well, here's what I think I can do. What can you do with that? So I think it's starting to force that. So those, to me, are sort of the big issues we've run into, or we're running into, with bringing renewable materials and into the marketplace.
And I think also the future looks more collaborative right now, and we will need more companies to start cooperating. Probably not just competing, but also co-creating and co-developing new products and technologies. So this is also really, really interesting, and I think it gives us really great insights on the future for this kind of molecules. And I will jump right now to Marilene, because you are working in some parts of this value chain, right? Like from carbon capture to carbon recycling, and also through the production of the manufacturing, the actual use of metabolic pathways to produce molecules. So tell us, how are you working, and also if you are partnering with some companies to co-create and to develop these new products.
Yes. So for us, it's part of one of the pillars, let's put this way, for us to get to success, is to have these partnerships. And this is for each one of the steps that I've mentioned before. So for synthetic biology, for example, we have our partners that can be considered more like clients. They come to us and they tell us specifically what molecule they want to produce. And then via what we know about the bug, and also with the help of the machine learning team, we can say, okay, so we are confident that we can produce this, and then we become partners. We have partnerships with universities as well, basic research, especially in our case, dealing with this non-model organism that we still have. It's a continuous process of developing genetic modification tools to work with it. So we have these partnerships, as Dan mentioned, our paper with Cell-Free, with Northwestern University. We have partnerships with universities in Europe, in Queensland. So this is a huge part for us. Our lean system, for example, the system that designs our experiments and help us build them. These are also being developed with a partner. For gasification that I said before, this is also with partners. We don't have all the expertise. I would say almost in any of the steps, we have 100% of expertise and capability capability to build and to produce efficiently what we want to produce. So partnerships for us are paramount, together with, of course, investment that can come from these partnerships or from the government or from these policies, which are very, very important for us as well. I would say that I say with confidence that there's no way to get there like in a commercial scale without partnerships. So that's paramount for us. Since, again, synthetic biology up to the commercial plants, we are partnering with someone that knows better how to do it, or has more capability or more money to do it. So yeah, for us, it's one of the pillars.
Thank you. And I will get back to this industrial part in a minute with both of you. But first, let's go to other part of the value chain. Because as Dan and you were mentioning, we have this set of tools, and we have some molecules that will serve as feedstock, and we have these molecules that are the products. And on the other hand, we have a great set of genetic resources hidden in the Amazon, right? So we can just imagine all the things that we could produce, getting inspiration and getting tools from all the microorganisms and plants and animals that live in the Amazon rainforest. So what is your vision on that, Guilherme? I've seen some efforts. The Inter-American Development Bank is also leading some efforts to promote bioprospection, to identify new molecules, to identify new targets, to identify new metabolic pathways, new non-model organisms to work as chassis. What are you doing in that field? And how are you contributing to this very smart use of our biological diversity?
Yes, I'd like to say, Daniel, that a lot more movement was happening, but the truth is that the initial movement is more on protecting the forest, which is a necessity, and also in helping in the improvement of the lives of the people who live in the forest, especially
I think we lost Guilherme for a second.
native people, into protecting traditional people. Sorry, can you hear me?
George, we'll let you for like 5 seconds.
Okay, so obviously a lot of effort needs to be input into protecting the native people, the traditional people in the forest. And you don't need a whole lot of technology. You need to get things done that we know how to do it. But what comes next? And that's where I think we are as a company, Vale is doing a lot of that. But as a research institute, we're helping to map the socioeconomic status of these people, but also we are looking into what's the next wave. So initially, we started our work to understand the plants that are impacted, plants and other animal species that are impacted by the company activities in the forest, because we are not, it's a net zero game. We're not going to lose any species while conducting the industrial mining activities in the forest. And there's a lot that is not known still. I'll give you one example. On the altitude fields where most of this work occurs, when we started the description of the biodiversity in the region, 200 plant species were known to inhabit this area. Then we combed it with specialists, over 100 specialists to describe the plant species, and we ended up with over 1,100 species. So this is pretty much the number of species that inhabit the entire United Kingdom. It's a lot of biodiversity that we still don't know. And while doing this work, we bumped into some species that are of interest, for example, for the agriculture and food production. So we have a lot of potato relatives. And FAO has already indicated that the understanding of the genetics of the natural relatives of crops is very, very important, because these crops will have to face increasing pressure from climate change and attached to it the new pests. We see that happening in Peru now, where the pests are going up in the mountain as the weather gets warmer. So the plants that are now native in biodiversity may have the answer for that. And one of the ways of translating that genetic innovation into the crops is through genetic manipulation. Obviously, there are other ways. But we ran also into some interesting situations. So one of the plants that we identified and helped to protect, it is an endangered species, but it's currently the only source of one drug that's used for treating high pressure and now to treat presbyopia. So the plant is endangered because the world needs this compound, this active molecule, but we cannot produce it endlessly by harvesting leaves from nature. So we developed an entire program into understanding the biology of this plant, into understanding, learning the genomics. What are the genes? What are the genes that may be involved in producing this compound? What are the intermediate compounds that are being produced? And that's done through transcriptomics, metabolomics, proteomics. And so we're throwing everything in to understand the pathway to get plants that produce this compound outside of the forest, but perhaps also to use synthetic pathways to produce this compound that's under extremely high demand globally now. So this is one example. Now we have a lot of unknown species there. We have a lot of species that produce compounds that we know as products that are not purified, that are used now by the cosmetic industry or by in the food industry. Now we need to understand which are the active molecules and how are they produced in order to translate that resource into something that we can use. And the global demand for plants like acai has increased the demand substantially. The price is sky high, and the consequence of that is that the local peoples have a hard time accessing it. So there are other ways of doing this. We need to improve the production of these species, but for no species in the Amazon, not even for the production of chocolate, we have a complete grasp on the genetics of these species. So we need to do that, and that is urgent. Urgent because we need to produce more, we need to produce better products, and we have the impact of climate change that nobody really knows what's going to happen with these species. So we can use from chocolate compounds, for instance. We know there are active molecules there that are of interest for the pharma industry. But so that's how we are viewing this, and that's why we launched this large-scale project to understand the genetics of the native plants, many of them which are already being used for the production of agricultural products, and developing those, but we don't want to redo what people have done with soybean production. This is going to be on agroforest systems, because this is how we do that sustainably, especially in the Amazon region. But we need to train the young generation to come in and be able to do all this work. We are just starting, and without you guys who are listening here, we cannot do this. So that's why we are here with all of you, to see if we can get people stimulated and gear up to work with us in this process.
We'll get back to that point towards the end of the discussion, because we have a gap that we need to fill in terms of training, education, and workforce development. But first, let's get back to the industrial part. So, Marilene, you were talking about the automation and the importance of automation. We know that you're working in this automated genetic engineering pipeline. So tell us a bit more about that, and what are your main accomplishments in that field?
Yeah, I guess for us, the main accomplishment is that, again, we work with an anaerobic microorganism. So everything, it's like strict. So no oxygen is allowed, never in the process, in any quantities. So one thing that we did was to build this anaerobic biofoundry. It is the first in the world. So it's a lot of different robots and robotic arms for liquid dispensing, for nano dispensing, for microfluidics, sealer spillers, incubators that are fed, that we feed with CO only, and they are connected to this biofoundry. Colony picking, transformation, everything we do inside this biofoundry, and this biofoundry is inside a gigantic glove box, so to be oxygen free. So is really something. It's really something new in terms of automation and high throughput screening and genetic modification of these microorganisms. So I would say that in terms of automation in our lab, this is the main accomplishment so far. We have good results with cell-free as well. But in terms of uniqueness, I would say that this anaerobic biofoundry is, and we are deploying another one this year as well. So yeah, we can modify thousands of microorganisms at the time. So it's really something. It's really something that sped up our process and made us more efficient, for sure.
And that's the way to go in the future. And I think it's I mean, this is going to accelerate research and development, but also we will need some more skilled and talented people working on that field.
Definitely. This is something that is already a bottleneck for us. And not only in automation, but automation is one of the main fields lacking this knowledge. But just synthetic biology in general, this is not a problem only for Latin America. It's not specific. So even in US and Europe, that you would assume that there are so many people in this field, there's actually a gap, and it's pretty worrisome.
So we've been working with some companies like OpenTrons also to start promoting this idea of automation, and that they have these also bold statements that we won't be pipetting in maybe 5 or 10 years. So that's not a skill that we should be developing in students right now. But it's kind of hard, right? Because we got to look at the future, but we also need to be here in the present.
We are in the intersection, yes.
and some of the young people listening in the other side, I think you should be concerned about this and start learning about these new tools and not just the regular lab work, right?
Right.
And well, getting back to Dan, you just opened a new facility in the US, right? They're in Boston. So tell us more about the plans that you have in Braskem to keep expanding your capacities and your facilities around the world.
Yeah, certainly. We're not quite open yet. So, but yeah, we
almost.
yeah, almost. So yeah, so it's interesting. We've been operating an R&D, biotech R&D group in Campinas, Brazil for at least about a dozen years or so now, right? And moved into our current space in Campinas, just right next to the sort of in the Unicamp area. It was about 8 years ago, I think, that we opened up that space there. And you know, I would say, you know, what we see is the team we have there is phenomenal. We have an absolutely amazing team, and their ability to drive products forward, or drive projects forward rather, is really impressive. I think where we run into issues or where we run into challenges is the availability of some resources, right? So the one I always point to is the availability of oligonucleotides, right? Just to get a primer can take 10 days, right? Versus here in Boston, you know, it's a day, unless I maybe missed the 2 o'clock deadline, right? And so I think that's a challenge, and it's something we continue to look at and what are the opportunities? Can we work with groups like Sinai and others to start to have, have some of these resources available in Brazil for that team and for everyone else? But I think in the meantime, what we're looking at doing is how can we essentially build a global biotech team that that basically harnesses the strengths of the various areas, right? The strengths of the talent pool, the strengths of the resources, et cetera. And that's really sort of formed us or pushed us to where we are today. So you're right, we're building a new lab here in Boston. It's going to be a beautiful 35,000 square foot lab space in Lexington, Massachusetts. And really, sort of going off what I was saying before, the real push for this space is to have it be multidisciplinary and be highly collaborative. So we will have, sitting in the same space, we'll have lab space for biotech people, we'll have lab space for catalysis, and those labs are right next to each other. If I'd had it my way, they'd be one lab, but for safety reasons, we had to partition them a little bit. And in the office space, it'll be catalysis, biotech, process technology, polymer science, open innovation, all sitting together. And the idea and the way we've designed it is to force them all to bump into each other and have lunch with each other and collaborate, right? So that's the whole purpose of the space. And from a biotech perspective, well, from an entire facility perspective, the focus is on renewable chemistry, right? And again, it's that agnostic approach to it of it's biotech, it's catalysis, it's some kind of combination thereof. We don't care. We just want to get there and develop renewable technologies that are good for the world and good for Braskem as we go. On the biotech side, I think you're going to see a lot of the standard stuff you see, right? We will have some automation. We will also have plenty of bench space for people to work at. I believe automation is a key component of the future, but I also don't think it's the end all be all. I think people will still be working pipettes and doing these kinds of things for the foreseeable future. Maybe I'm old fashioned. We'll have some small scale fermentation capabilities, but really the purpose of the team, the goal of the team is to take technologies from an idea up to about proof of concept. So if we're thinking technology readiness scale, we're thinking 3 or maybe 4. Take those technologies once they've reached that point, and that's the stage where you want to be able to turn things over quickly, access to certain resources very quickly and inexpensively will be really critical. So we're harnessing what's good about Boston and this area. Those technologies will then transfer to the team in Campinas, and their job will be to take it, pull it up, do further strain modifications, still do a lot of that strain engineering, synthetic biology work to bring the strains up, and then we'll start doing the fermentation work and push it all the way out through pilot scale, and then go ahead and commercialize those technologies from there. So we're going to have 2 campuses, a single team, and we're going to have that transfer as technologies mature. Again, harnessing the strengths of both areas, right? And that's how we see it all sort of coming together and what the focus for the team is going to be. Our goal today is to have that space open in May of 2024. Supply chain is a real challenge for everyone, and some of the equipment for the building itself is delayed significantly. So.
And this is going to be a really wonderful model for cooperation between the North and South, right. Between the US and Latin America, right? So, and precisely about that, we've seen that this education gap, this training gap, is also a thing in Boston, the world capital of biotechnology, where probably 70 or 80% of the biotechnology of the world, or at least from the American biotechnology, is held, and they are having issues getting trained people, skilled people to work for the company, and this is also happening here in Latin America, of course. So the final question for all of you would be: how can we fill that gap? How can we contribute to train more young people to kickstart their careers in biotechnology and synthetic biology. And what would be the vision of your as individuals, and also the work of your company, or the concerns of your company, probably at this stage. And I will open the floor for whoever wants to start with this last round.
Okay, I can kick off things. So first thing I think we need to motivate people to do this. We need to train people, but we need to train people that have a purpose and a reason and motivation to do this. And I think we are in the Amazon region for a reason, and the reason is to make sure that we use the natural resources in a responsible manner. And we are willing to, the company has put nature-based solutions as part of its strategy, not only as a possible source of revenue for the company, which for Vale has to be something fairly sizable, but it can translate into the benefit of other people who get jobs and startup companies. So we looked at the biodiversity currently, the potential for the use of forestry products in all sorts of ways is for about $4 trillion according to McKinsey. Currently, the Amazon forest responds for 0.17% of the global production of extracted forest products. That's absolutely ridiculous, and that's mostly being used to produce very low price values. So if we look at the biodiversity we have, and we see the potential that is there to protect this biodiversity while making a business out of it and producing things that society needs, not another fad, but something that we actually need, like the products that our colleagues have described. I mean, if this is not, I think this is purpose enough for a lot of people to get up in the morning and prepare themselves for the next wave. And what we hope to do is to do two things. One is to participate in the movement that iGEM is conducting, to get people motivated and understanding that this is a very interesting way forward. And we need people globally, much even more so in the region. And also we are trying to position and move the entire system towards that by doing things locally. I completely understand that it is a challenge to get, I mean, we live that, to get things in the country. Oligonucleotides is one example among many. But I think that as we start creating movement around here, we will be able to get these products in a more timely manner, and in a more in price-wise, something that's at pair with what our colleagues obtain overseas. And we are connecting with everybody here is talking about bioeconomy with many different flavors. The flavor that we have here is we need to is a biotech bioeconomy that will come in as a next wave. And we need to train people, we need to create an environment, and we are working with the state government to create something very similar to what Dan said, a place where we can get people coming in and out. We collaborate extensively, it is open, and I think this will be a very fertile ground for the young people to come in and work and get their startups up and running, and that's where we position ourselves. And thank you for the opportunity here, Daniel and Carolina, and for learning so much with Mari and Dan.
We are also learning a lot, and you mentioned some really interesting facts, and it's very important also to start getting people from outside the capitals, not just Sao Paulo, Rio, Mexico City. We need people from Belém, people from Manaus, people from Mendoza in Argentina, from Talca in Chile, from Santa Cruz in Bolivia. So what do you think, Dan and Marilene? What's your vision on this?
I was going to use the word enforce, but I'll change to incentivize. I think we should, and usually these kind of incentives, they come from the government. So, for example, it's controversial in my opinion, but I really like the model that the US have for some grants where they put an educational component attached to them. So if you receive a grant, you have one of the requirements is that you develop an educational or workforce development component to it. And I really like this model because it's something that is an incentive that is both political and also financial. So if you're getting this money to do whatever you want, to produce whatever you want, or develop the technology that you want, so you have to attach to that, transferring this knowledge to the new generation. And I really appreciate it. So I guess this kind of incentives are, in my opinion, are very powerful to get us going and actually have a plan and have projects in place to train the new generation, to train, and not only the new generation, because this technologies, they change so fast that we ourselves already work in this field will have to be trained eventually in something new. So yeah, just to develop not only the new generation, but the workforce in general, I think these components are a good incentive.
We need to keep learning every single day. I remember when I finished my career.
Guess, yeah, and I'm not complaining. I love it.
No, it's perfect actually. It's super interesting to. I'm feeling like a bit rusty.
Yeah.
with the new technologies coming up. And yeah, let's close this round with Dan. What do you think?
Yeah, I think I tend to agree with some of the comments that both Guilherme and Mari brought, where, you know, I think there's no silver bullet, I think is the answer, right? It's going to take a lot of different approaches here. I think, you know, one is yeah, how do we get the government to incentivize education, right? You know, and I think another big one is access to resources. How do we speed up that access to resources? And to me, it's going to take things like the government stepping in and saying, look, we're gonna fund a project to build this capability, you know, in Brazil or wherever else maybe. Again, I'm very Brazil centric just because it's where my team is. But I think it's going to require some major sort of push on the government to figure this out. I'm hopeful that you know that's something that can happen. You know, beyond that, as far as thinking about, you know, what is what would I like to see the synthetic biologists of the future, what skill sets? I think one of the biggest ones, and we've talked about it again and again, is the ability to collaborate, right? I have plenty of really smart people who are incapable of collaborating really well. And you know what? That's a dead end. That's not useful to me as a leader. Is you got to have people that are able to collaborate. So I would push everyone, you know, young scientists, find ways to collaborate, find ways to work in multidisciplinary teams, learn how these other teams work and really understand that. I think, you know, the other thing is, and Mari pointed this a little bit, is you better know how automation works and how some of these other things work. But the other shortage I'm seeing a lot of are systems biologists, right? Sort of our kissing cousin, as it were. You know, it's a very similar field, thinking about pathways, thinking about pulling genomics and other omics together, and pathway prediction. I think it's still a major hole that we all have in front of us. And absent that, you know, we can't go build the bugs and the tools that we need to build. So those are the things I would love to see more of a focus on from an education standpoint going forward.
I love this conclusion, and this is something we totally agree on. We need to train and to push collaboration. Curiously, in IGEP, we kind of force the teams. It is a mandatory requirement for them to collaborate. So you need not just to collaborate with a multidisciplinary team, but you need to go outside and collaborate with your competitors.
Yeah.
So this is a very, very important value for us. And let's close. Thank you all for your really valuable contributions. I will ask you to close with a conclusion, but it's going to be a tweet. So a very direct and straightforward conclusion. So while you think about this, I will thank everybody here in the audience for your presence and for your attention. We will keep having this kind of events. We will be talking about health. We will be talking about food, also about platforms. And we're interested also in having some in-person events. Carol is going to tell you a bit more about this. We're thinking of going to Rio de Janeiro, to Monterrey, Mexico, here in Guadalajara, probably in Buenos Aires, in Bogota. So we want to keep this conversation going. And yeah, before going to Carol, I will go with our panelists for this conclusion. So tell us, what is the key for this transition to a sustainable bioeconomy from this point of view, according to all the things that we just mentioned? Who is the bold one to start with the conclusion?
I would say that is investment. We definitely need investment. Either if it's for infrastructure or for developing the new workforce, to develop new technologies, supply chain, we need investment. So, and usually this investment, of course, they can come from private institutions, but for disruptive innovations, I truly believe that the government has a big part of it. So, yeah.
Totally agree. And we need to invest not just money, but also efforts and willingness and passion in this. Who's next?
I can go.
Go for it. No, please,
okay. Yeah, I think obviously investment is essential, but I think it needs to be directed. And we see here in the Amazon region, everybody wants to do something here. And there is already a lot of money, but it can be all over the place. So we need to have a strategy to make this investment come to fruition in things that we all believe are really important, like synthetic biology as a way of generating value out of our biodiversity.
Totally agree as well. And finally, Dan?
Yeah, so again, I agree 100% with Mari that investment's a key part of it. And I think within that, I think federal investment, so government investment, is probably more appropriate given the timescales we're talking about, right? I think this is something that the biotech industry has struggled with, especially when we're talking commodity chemicals and fuels, is that the investing space says we want to see something in 5 years, and unwittingly we say 5 years, that's fine, we can do that, and then 10 years later everyone says, well, where is it? And we're just not talking those kind of timescales, given the still the relatively early days of biotech, right, for these types of chemicals. So I think I would like to see more federal government investment. And Guilherme's point is a really good one of targeted and appropriate investment. Here in the US, we've started having these programs called Biomade and Remade, and these sort of centers. And essentially what they are is the government gives a bunch of money to get these things up and running, but then they have to be self-sustaining. I love that model. It's a fantastic model. I think the other thing I look at, and I'm a bit of a broken record here, but it's collaboration, and it's collaboration across companies. It's collaborations across industries, across academia, industry, startups. I think all of these guys, all these companies need to, or all these bodies need to find ways to work closely together. And it's not, there's no silver bullet, right? I mean, I absolutely love Lanzatech's technology. It's one of the most, earlier when I was thinking about what's exciting, it's one of them, right? But I think, that's not the only solution. what we're doing here at Braskem is not the only solution. And so we need to find ways to work collaboratively to drive these things forward. So that to me is one of the other big ones, and then education. It's just, we've got to train the next generation of scientists and engineers to be ready for the challenges that are in front of them. So
Totally agree. So education, collaboration, investment, and strategy are the keys for the future. Well, thanks a lot. You've been wonderful. I've learned a lot here. And well, thanks also, Caroline, for putting all this together. Amazing work. And yeah, you have the floor to close the session.
Thank you, everybody. Thank you, Marilene, then Guilherme again. Well, I would like to say thank you for everyone who is here, who attend to this event. And I would like to invite you to get to know more about our work here at iGEM Design League and how we are developing projects to empower Latin American students to design with biology. But also, we are organizing more events like this. So soon we are gonna send you NPS through email, please answer. But you can tell us if you wanna get in touch or maybe partner up with us. We can develop a project or work together to empower students and to expand synthetic biology here. So this is my quick message. And the next event will be in person in Rio de Janeiro, along with SENAI, the Institute of Biosynthetics in Rio de Janeiro. So we're really excited to meet the community from Rio de Janeiro, of course, from Brazil. But in June, we are planning another online event. So be in touch with us and following us in the social media. So that's my invitation for you all. And thank you.
Caroline?
Hey.
Sorry, when is going to be the event in Rio de Janeiro?
It's going to be on May 16th.
oh,
Okay. But we are still like closing the gaps and we are going to announce everything soon.
Okay.
But you're all invited then, Marilene, if you are in Brazil, of course, and Guilherme, if you are in Rio de Janeiro, you're super welcome to the event. So thank you.
thank you, everyone.
Thank you. Bye-bye.
Thanks. Bye-bye, folks.
Bye-bye.
Thank you for coming.